In this video, Dr. Francis discussed the use of Agricola Package in a breeding experiment, as well as providing steps to install the package into your computer. He also describes an application of alpha lattice design in a maize disease experiment.
We're going to be using the Agricola package. We're going to use it for the randomization of our treatments, for the analysis of variance, and to retrieve the adjusted treatment means. So this package supports quite a few other functions beyond what I'm going to talk about, and I just want you to be aware of these. We are going to use the design lattice function today. We're going to use the PIBI test function. We're going to get F tests out, but we're not going to do mean comparisons. We are going to do some data visualization. The package works in conjunction with mass. In early versions, you had to bring in mass independently. I think that it's set up as a dependency now so that you don't have to do that. Let's go ahead and make sure that we have Agricola installed on our computers. So you can see that when you bring it in originally, it is bringing in a number of dependencies, unpackaging them and checking them. So now let's talk about the data that we're going to analyze. Maize lethal necrosis, MLN, is a disease that's caused by combined infection of maize chlorotic mottle virus and any of a number of serial viruses in the potyvirus family. So sugar cane mosaic virus, wheat streak mosaic virus, or maize dwarf mosaic virus. This is a disease that has been spreading throughout sub-Saharan Africa and can be devastating. It's definitely insect transmitted, but also may be seed-borne. And so anytime you get into a seed-borne disease that does this kind of damage in a field, you get a lot of finger pointing, you get a lot of the bureaucracy involved, and it has the potential to be a mess. So you get countries that say no more importation of corn seed, even though they may not have a seed industry established. This is the kind of disease that has the potential to lead to starvation in countries. Okay, so there's a lot of interest in trying to find resistance that will help ameliorate the problem. For now, all African maize germplasm is susceptible to MLN. Victoria Bulagaya, who's a master's student here at OSU, she's part of the iAgri project, which is a cooperation with Sokoine University of Agriculture in Tanzania. The goal of her study was to test whether potyvirus resistance QTL would work to reduce MLN symptoms, and then assess which allele combinations would provide resistance. The justification for this was that we already knew from Jose Luis Zambrano's studies that there were genes that were effective against the potyviruses that contributed to this disease, QTL. The most important ones of those mapped to chromosome 3, 6, and 10. Resistance to the other virus involved in this have not been published. Okay, so the question that Victoria set out to address was whether any of the existing QTL or combinations of those QTL would be effective at limiting disease. So this slide summarizes Jose Luis's work, and it shows those QTL on chromosome 3, 6, and 10, which were effective against the potyviruses that are highlighted in blue. And so what Victoria did is she went to Jose's population, a large rill population, and she selected lines, 5 lines that had the chromosome 3 QTL for resistance, but not the chromosome 6 or 10. 5 that had the 6 but not the 3 or 10, 5 that had the 10 but not the 3 or 6, and then 5 others that had both 3 and 6, 5 that had 3 and 10, 5 that had 6 and 10, and 5 that had all 3 of them. So she knew she didn't have the resources to run the whole population, but she selected a set of lines that would allow her to address whether specific QTL are important. So why 5? 5 comes out of theoretical paper that describes bulk segregant analysis. If you have 5 in each group, the rest of the genome is randomized around those. So you're really, if you treat it as a group, You've isolated that region and you're asking, is this region important? So that's her genetic material. The data I'm going to be talking about were data that she generated here using growth chamber experiments. She's replicating this in the field now in like 5 locations. Okay, the growth chamber experiment, she was evaluating the effect of these pottyvirus resistance QTL on MLN. So it's an artificial inoculation with maize chlorotic mottle virus and sugarcane mosaic virus. 35 selected lines from the real population. I've already described this. So each of the 5 treatments has alleles for pottyvirus resistance from the 3 QTL. 2 parents, 5 resistant susceptible checks, so that's a total of 42. Inoculation, 10 days after germination, inoculate twice, and then begin scoring after symptom observation.